Welcome back. In the last vignette, I talked about science is not a smorgasbord. That was several vignettes, but the last piece of the last one, the scientific content, was the idea that over the last 150 years, we learned about the existence of pathogens, and out of that emerged what I am going to intentionally refer to as the germ theory of disease. I wanted to now just say a few words about viruses and how we came to know of them, and this will serve as an input into our discussion of viral evolution, but also physical virology. How do we think about the physical processes that attend being a virus? And there are many, in super interesting ways, and I'll give you a hint of that in this particular vignette. So in the 1890s, a new class of pathogens was discovered. They're known as viruses. It took I would say something like 30 years for this to really kind of cement into the modern picture of biology. And what I think is very interesting about this is that it was really the work of two very distinguished and excellent, I'm going to say, microbiologists, Ivanovsky and Beyerink, and they were confronted with disease in the same way as had been Pasteur. And what I mean by that is that Pasteur, he had all sorts of industrial interactions. People came to him with a desire to better understand what you might call contamination in processes of fermentation, for example. So there were both breweries of beer and wineries, vineyards that came to him. I don't remember the details off the top of my head, so I probably don't have this historically correct, but I do have the concept correct, which is that he was interacting with these industries and trying to understand what was going on when the fermentation process didn't work out the way it should, and out of that, he discovered that there were contaminants, microbial contaminants. And in fact, there was like a trajectory in his mind from that to human health, because he thought that things like the putrefaction of meat, that maybe when we get sick, that we should think of that as a kind of putrefaction of our own tissues. And the idea being that somehow we had been invaded by a pathogen, which up until then was thought of as being what we would now call a bacterium. So recall I told you about Vibrio and Vibrio cholera, and about Koch's postulates. And Koch's postulates, just as a reminder, are the idea that if you really want to demonstrate that you found the causative agent of some infection, then the way to go about that is to culture the infectious agent from the infected animal, to grow them up, and then to reinfect, and then to reculture, and have that whole cycle be performed in a way that's compelling and convincing. So these guys were confronted with disease of the tobacco mosaic virus plant. Very interesting. Which leads to this splotchy behavior that you see on the right. And you might have imagined that the way that you would go about trying to find the causative agent would be to use filters. And what I mean by that is, like, if I use 1 micron square type filter size, then if I were to take an infected plant, to remove all the juices, let's say, get rid of the plant material, and then flow that liquidy flow through a filter, what I would expect is that the bacteria would be left behind. And that's generally what happens if you have a bacterial infection. On the other hand, what these guys found, both of them, sort of simultaneously, is that, in fact, the flow through itself remained infectious. And this led them to posit the existence of a new category of infectious agent, which was down in scale, let's say, by a factor of 100, or maybe a 10, between 10 and 100, something like that. So, a virus can be as small as maybe 10 nanometers across, and maybe as big as something like 100 or several hundred nanometers. So SARS-CoV-2, the one that's been plaguing us for the last 2 years, has got a size of about 110 nanometers. So these guys then posited the existence of viruses, and now I want to tell you about work that took place, a solid 60 years later, and this is one of my favorite all-time experiments. This is definitely in the variety of experiments to change your life for. There's two of them that go together. So in Berkeley, what they were interested in was, could they reconstitute an active tobacco mosaic virus from its components? And the thing about TMV is that it's very simple. And when I say simple, I don't mean that. I mean, it's simple and it's very complex all at once. It's simple in the sense that there are not very many components. So what they were able to do was they were able to purify the proteins. So this is an RNA virus. I'm not sure. Let's see if I can actually remind everybody the story. Yeah, this will be useful. So just as a reminder, there's different ways to classify and think about viruses, and I think the best classification actually is the one that was introduced by David Baltimore, the former president of Caltech, and actually somebody that I've collaborated with. And he had a very cool classification scheme, which was, is it RNA or DNA genome? And then if it's RNA or DNA, is it single or double stranded? And if it's RNA, is it the positive or negative strand? And that kind of stuff. And here I just wanted to show you, all these different categories of virus. So tobacco mosaic virus, which I guess I didn't put here, is an RNA virus. And so at any rate, they basically were able to isolate the RNA. They were able to isolate the proteins. And then, as you see on the right-hand side, beautifully, the viruses assemble into the viral proteins assemble into beautiful cylinders, but they have a distribution of lengths. And that is in itself fascinating, because the infectious viruses have a very uniform series of lengths, and that's what led to this amazing 1977 experiment that was reported in PNAS. So what they found is that there's two little fragments of RNA that are hanging out of the nascent, growing tobacco mosaic virus. And what they did is they measured the length of those two tails. And what they conclude is that you can see the long tail keeps getting shorter and shorter and shorter. And in a way, it's serving as a tape measure. What I mean by that is that the addition of monomers, protein monomers, to the cylinder continues to go on until the template, which is that long strand of RNA, has been exhausted. So, the graph in the middle, this one, shows us the length of the long tail, and on the right is a cartoon that shows the temporal evolution as more and more monomeric units are added onto the TMV capsid until finally the whole thing has been assembled. People did mutants where they changed the length of the genome, and then what happened is that the assembled virus was shorter and that kind of stuff. So this is a very, very beautiful example of sort of the unification of in vitro biochemistry methods and deep biological things having to do with, how do living organisms and how do viruses measure and control their size. So this gives you a very concrete example. So, the point of this little digression, this little tangent, was just to, it still fits into my general theme of science is not a smorgasbord. It's really, really unacceptable to me. It's a very subjective comment on mine, but it's unacceptable for us in this modern day and age to go to airports and to climb on airplanes and to live in a world where between 2009 and 2019, I think there were 0 civil aviation deaths of U.S. airlines. I mean, it's, 0 airplanes, big jet airplanes crash. 0. 40,000 Americans die every year in their cars. So, we're talking about people going into the third dimension. We send satellites, off into the space. And so we know things in science. Do we know everything? Absolutely not. Are we confused? Absolutely. We're confused all the time. But there are definitely things that we can bank on. I know I can look at the tide chart and go down to the beach today, and the tide is going to be what the tide chart says. And so we know about viruses. It's not some sort of a fantasy. It's not some sort of a conspiracy. And furthermore, viruses are dangerous to human health. I think that's not a controversial statement. And so, here you see a cartoon of Ebola. All I can say is that I for sure would, if I were in an Ebola zone, I would be wearing one of those big suits. I can assure you that. And I really wouldn't want to be there in the first place. Anyway, so that was point one, but then I also just wanted to, meaning, it was point one was science is not a smorgasbord. We know about viruses as infectious agents. We know a lot about viruses. We know beautiful things about their structures. I mean, look, here's some electron microscopy studies done in Grant Jensen's group where they looked at HIV, its heterogeneity, and so on. Really, really cool stuff. We know a lot about viruses, and I just wanted to say, a little bit of the narrative of how they became like an addition to the original notion of the germ theory of disease, which is very cool. That's an example of how we broaden our knowledge. We got to a point where there was a notion that certain bacteria are infectious and can cause human health problems, and there was another kind of infectious agent that was discovered, and that's the so-called viruses. So what we'll do next is I'm going to talk about evolution of viruses, and I'm also going to talk about physical virology.